Sure. Thanks, guys, for having me on the show. My name is John Gill, and I'm a senior director here at CODEX DNA in the R&D group. And I've been working in the genomics and synthetic biology field for just over 25 years now. Started out at the Institute for Genomic Research in the late 90s. Was involved in some of the early genome sequencing efforts there that were done, as well as the first haploid human genome. And then in 2011, moved over to Synthetic Genomics Incorporated here in San Diego. And I've been working with Dan Gibson on various projects, the Digital-to-Biological Converter, which I'm sure we'll cover, and more recently focused around synthetic or enzymatic DNA synthesis for synthetic biology applications.
So could you actually give us sort of, would you say maybe an overview of the key technologies that you're working on? And then we can sort of delve a bit deeper into each of these.
Yeah, so probably, I guess, starting in 2011, there was a concept that we came up with called the Digital to Biological Converter, or the DBC. And the idea behind that was to come up with an automated system that could take in digital DNA sequence and make the oligos, effectively pool the oligos to make genes, and then output therapeutic targets potentially. And then what sort of spun out of that was a system that we now sell, that we have on the market, that we've had on the market for a while now, called the BioXP. And what the BioXP does is it takes DNA oligos that are already pooled into microtiter plates and assembles the oligos into genes. And then it goes through an enzymatic DNA error correction process that effectively removes a lot of the errors that are often in the oligos. As a lot of people in synthetic biology will attest to, there are quite a few errors in traditional oligos made with the traditional phosphoramidite synthesis process. So we had to devise a way to effectively remove those errors, and then the output from that system are full-length assembled genes, and they can go into several applications that we also have automated on the system. Gibson assembly can clone into your favorite vector. We can also go into a process we call rapid amp, which we can amplify lots of DNA for transfections. And more recently, the system is also producing mRNA. So if you want to screen some potential candidates for mRNA vaccine research, you can do that on the system. And then probably in the not too distant future, maybe the next couple of months, we're going to be able to make protein on the system as well. So that's sort of where a lot of the focus is here at CODEX DNA. That's our sort of flagship product, and what my team works on here, and other teams as well, in terms of manufacturing the reagents, product development, marketing, commercialization of all the products that we have. Yeah, and then we have some other, we sell Gibson assembly kits to customers, as well as a cell line called VMAX, which is sort of like an E. coli replacement that can propagate colonies pretty rapidly and kind of cut down on the design, build, test cycle. Because if you can get colonies within the same day that you transform, your clones, it can really help.
How did you go about reducing the errors in the oligosynthesis?
So on the BioXP, we use an enzyme. It's an error correction enzyme that we discovered in about 2012, I believe, and it's a single enzyme that can actually recognize sort of heteroduplexes, so errors effectively in the DNA. And that's a pretty robust enzyme at doing that. And it's sort of unfortunate that we have to find an enzyme that can actually, eliminate errors that are in the DNA. But despite the fact that traditional phosphoramidite chemistry has been around for over 40 years now, it's still to this day we have issues with oligos having errors in them. So we had to devise this enzymatic method to eliminate those errors.
Okay. What is the BioXP? Is that a machine or a method?
Sorry, yeah, it's a machine. It's an automated platform that's specific to building DNA starting from oligos and then error correcting them. So the system has, multi-channel pipetter on it. It has a thermocycler. It has, space for pipetting tips. It has a magnetic base for purification of DNA. So it's sort of like it's a compact, we just think about it as a synthetic biology workstation. And we're constantly thinking of new ways to sort of devise applications that can go on, sort of be at the tail end of the gene synthesis. And of course, our customers that we work with, they help us, kind of push things to the next level. But yeah, essentially it's an automated system that builds DNA.
Right.
I'm curious about sort of when you were initially sort of starting to build this automated system. Where did you get your inspiration from, or how did you decide to how did you sort of design this sort of modular kind of system?
Yeah, it's a great question. I think a lot of the inspiration came from, some of the work that Dan Gibson was doing on the first minimal cell. Sorry, not the first synthetic cell. The minimal cell followed after. But one of the things that he and the team at the Venture Institute realized was that this process could be pretty cumbersome, and then all the errors that you have in the oligos. So if we still think the errors are kind of bad now, in our oligos that we get from suppliers. Imagine what it was like almost 15 years ago. The chemistry was still, after 25, 30 years or so, is still evolving and still not quite there. And we thought that having a system to sort of automate this entire process of building gene linked fragments that you could then even take and build into higher order assemblies, for pathways, genetic pathways, or even whole genomes, would be extremely useful for other scientists that were trying to do the same sort of things that we were trying to do. And it turns out that people really need this technology and want it. So we're spending now quite a bit of time on trying to build the front end of the system, which is effectively gets back to this idea of a digital to biological converter, where you have not only the BioXP assembling the genes, but the upfront process of making the oligos via enzymatic DNA synthesis method that we call SOLA.
So can you explain how this SOLA sort of works a little bit to, just describe how it works, yeah?
Yeah, absolutely. SOLA it stands for short oligo ligation assembly. So instead of building oligos in a traditional way where you add individual DNA bases at a time, this starts with a pre-manufactured set of very short oligos. And it has all permutations in this set. We can call it a library of oligos. And those short pieces of DNA are effectively coupled together, and they're sort of trapped between surrounding kind of conserved DNA. And this sort of trapping process gives us the ability to increase the fidelity of the DNA that we're trying to make. And then progressively over time, you go through cycles. It's 3 cycles of a 2-step process that we go through to basically unearth the DNA and kind of build progressively larger and larger pieces of DNA. So it's, I would call it a synthesis strategy for making DNA, but it's effectively an assembly process. It starts out with really short pieces, and because these pieces that are in the library, it's not like it's a million of them. It's not, you can have the entire library set on an automated platform. And that's kind of what we're focused on right now, is building an automated system that can effectively build these short pieces and go through this assembly process, using entirely enzymes. They're we're not producing any hazardous chemical waste like the traditional phosphoramidite chemistry does. It's all through enzymes and using very dilute amounts of these library components.
I'm really curious. So because I feel like a lot of people are not a lot of people, but some people might be sort of in some ways afraid of taking on automation, especially when it comes to making creation of experiments or things, or using automated tools. Maybe you have a bit of hesitancy towards automated tools. How do you how do you ensure that using automated tools is sort of the errors that occur with using automated tools are also accounted for?
Yeah, so the one thing I think about these tools, like the BioXP, for example, so it is a system that you have in your lab. It's not, in a sense, a traditional automation system where you have to program all the steps that you need to do. So you're right, yeah, like you can introduce errors in that programming process. So what we go through is a series of, we go through a development period where we're trying to develop the application that we're currently working on, and that goes through a lot of sort of verification and validation to make sure that it works, that the hardware performs well with the chemistry. So I think the analogy would be like, if you had an Oxford nanopore sequencer, you're not programming these systems to do specific things in the lab. They're in your lab so that you can get your work done faster. So you can spend time doing other things, thinking about experiments, and not programming automation. So that's sort of how these systems are devised, and what we try to do with them. We don't want to put the burden on the customer to take time away from their research to program the system to build DNA. It's already, those workflow steps are already embedded in the product.
What size DNA fragments can you sort of construct with this method compared to the older method?
Yeah, great. So it's so far we built a number of genes, and recently we just had a pretty significant breakthrough where we were able to assemble the entire spike gene from SARS-CoV-2. So that's about 4 kb. So effectively it was taking, and the way we build the fragments with SOLA is that we go up to 100 base pairs. So we start with very small, and then we progressively get larger, and then we get up to 100 base pair. And we've learned that 100 base pair is a really good stopping point. And then we can take all the 100 base pair pieces, and it was about 70 of them for the spike gene, and they overlap each other. So they can essentially act like links in a chain, where they're connected in a certain way. And then through some assembly processes that we've developed in-house and that are actually on the BioXP, we're able to assemble the entire spike gene with the SOLA technology. So, and then when we look at the errors, even without error correction, we see really good error rates. So just to kind of put it in perspective, when you get oligos from traditional phosphoramidite chemistry, the error rates are about 1 in 500 base pairs. One error per 500 base pairs synthesized in your genes. So what we saw with this particular assembly with the spike gene was about 1 in 5,000, and that's without any error, any enzymatic error correction that I mentioned earlier.
Right,
Wow.
that's incredible. Yeah.
Yeah. Do you think that the system could be later maybe applied to construct? I mean, you mentioned that you have been constructing the spike gene. So do you think later on the system could be improved to also do it with like larger DNA fragments, like with sort of like assemble like proteins?
Yeah, absolutely. Trying to go up to, okay, so we could take the SARS-CoV-2 genome, for example, 30,000 base pairs. So we could imagine building, a number of 4,000 base pair pieces of DNA, and then employing technology like Gibson assembly to then put all those DNA fragments together to assemble, the entire genome. So I think once you get to a certain place with the DNA fragments, whether it's 1 kb, 2 kb, you can stitch them together to build, larger pieces of DNA. It's really the key thing is getting the starting, the starting oligos, the starting pieces of DNA. That's been the real problem. So once you can get those starting pieces made and you can make them in your own lab, and it's decentralized, then sort of the sky is the limit. You can make all kinds of things pretty rapidly, whether it be lots of different genes for protein coding sequences, genes to make up an entire genetic pathway, or genetic pathways to make up entire genomes. But it's getting that front end part to make the DNA oligos rapidly, cost effectively, and in a way that's safe for the environment, for the employees that are working with the DNA. And yeah, I think that's the game changer in terms of getting larger pieces of DNA is actually getting the small pieces done quickly and efficiently and cost effectively.
And how long does it take usually, say, to make that spike gene that you talked about? How long would that take? Or did it take?
So the way that we did that, it was through a semi-automated system. So we have, it's kind of broken up in a modular process, and we're moving things onto thermocyclers and putting things on automated systems. So it's not completely automated just yet. That's the goal, is to get there. But even doing it in a semi-automated fashion, it takes about 2 days to get to that gene. So if you try to do that in your own lab, you'd have to you likely don't have an oligosynthesizer in your own lab, because they're just dangerous to have around. They generate a lot of waste, and then they're expensive, and you have to dispose of that waste. So you would probably end up ordering the oligos from a vendor, and then you would need to get those oligos in and pull them together and then start assembling. So it could take, anywhere from 2 to 4 weeks, honestly, to do that. So yeah, we want to have this technology collapsed on automation platforms. Not very large systems. We're talking benchtop systems. We're not talking the size of one of the older PacBio sequencers or something. It's not an extremely large platform. We want to have this embedded in these systems that can enable synthetic biologists and folks doing biotech work and in their own labs, and potentially one day maybe even in the clinic as well, hospitals, probably down the road a bit. But yeah, I think that's sort of an aspirational goal for us as a company, is to get there.
Are
Sorry, go ahead.
Are your devices such as this, the BioPrint XP, are they already being used by customers, and how widespread are they being used?
Yeah, the BioXPs are being used by hundreds of customers, from from big pharma to startups to academic labs, core facilities. Yeah, they're pretty, they're not as common as as a thermocycler in the lab just yet, but we think they're getting there. We think they have the potential to get there one day. But yeah, all different kinds of applications, from building, screening CRISPR-Cas9 mutants to, at the front end of sort of like the entry point into the clinical therapeutics, and building whole genomes. So we've built a number of SARS-CoV-2, pretty much all the variants of concern for SARS-CoV-2 on the BioXPs and assemble them into full-length genomes at the bench. Yeah, they're pretty widespread use, and of course, as a company, we hope there'll be continued uptake of the system, especially when we can get to sort of this concept of a digital to biological converter.
You mentioned a little bit about protein synthesis. I was just wondering what sort of stage that research is in.
Yeah, it's pretty far along. I think it's going to be probably launched as a product in the very near future. It's definitely this year. It's going to happen. So that'll be a pretty exciting, I think, launch for us, because that'll sort of complete, all the different, producing the full-length genes, in the form of DNA, and then mRNA, and then just completing kind of the trifecta, I guess, of having the protein produced on the system as well.
So if you have sort of fully realized the potential of this product, what do you see it doing for like laboratories that are using this product?
For the BioXP, or for
I guess focus on one.
Yeah, so one example, I think, for if you're talking about BioXP applications, is we've had a couple of customers that are interested in building what we call neoantigens, and this is pretty close to the clinic, where patient samples, the tumors will be biopsied, and they'll actually sequence the tumor cell, and then they'll look for neoantigens, which are effectively SNP-based targets, where the proteins that are being expressed in the cell have some mutations that are sort of driving the progression of the cancer. So these are actually pretty small pieces of DNA. So we have some customers that want to take the patient samples, and then they want to make the neoantigen DNA to express that, potentially into protein. So it's getting like super close to the clinic, and it's very exciting, and it's very personalized, kind of targeted cancer care that really relies on DNA sequencing and DNA synthesis. So I think this is sort of like the kind of thing that's just really exciting to me, is to see synthetic biology getting super close to the clinic, so that you can potentially have a therapeutic that's based on making DNA, and that's sort of the same with mRNA vaccines. It's also very exciting because customers working on mRNA vaccines have BioXPs, and they want to produce mRNA to effectively screen for mRNA candidates for vaccines. So, and then all the way to, just basic research and people wanting to tighten their design build test cycles around, the research that they're doing. So instead of having to wait, 2 to 4 weeks for a gene, they can use the current BioXP system that we have to build genes within 5 days. And then potentially when we have, the enzymatic synthesis kind of bolted on, if you will, to the front of this current BioXP system, potentially be able to make genes in 2 days. So that's I think also really exciting in terms of what we want to do, where we're going as a company.
Yeah, that's great. Yeah, I think especially having ordered DNA from various websites and then not coming for
Yeah,
a month
yeah,
or two, sometimes it can be a bit of a pain.
yep, yeah. We hope to change that. It just shouldn't be, and it's frustrating because you see with sequencing how far sequencing has come along, with NGS in 2005, and you can have your own sequencer in your lab. You can do your work on your own terms. You can launch major experiments, sequence whole genomes, and you don't have to wait. So it should be the same way with DNA, writing and synthetic biology. It should be exactly the same way as that. Maybe it'll take a while it'll be mismatched for a while when you compare how quickly you can sequence to how quickly you can write, but I think that gap will start to close over time.
Yeah,